Okay, good morning everyone. Okay, good morning everyone, good morning. Okay, so Annie, try to watch the classes in case if you miss anything. Okay, so this 1 week please spend more amount of time in revision. Okay, so everyone try to spend friends, a minimum 10 hours minimum. I'm using the word minimum, and please try to read at least a maximum of 13 hours. Okay, even if you are really having a very good patience, you can take a peaceful sleep of 6 hours and you can spend more amount of time to study. What is the reason I am saying to read more number of hours? Please remember my words. Whatever the things that you people remember in the last 7 days, once again remember, whatever the things that you people remember in the last 7 days, you can easily recollect in the exam. Okay, that's good. Good, Annie. You can recollect easily in the exam. So that's why in the last 7 days, instead of reading the new topics and wasting the time, always try to revise the topics which we have already studied and try to recollect it multiple number of times. Okay. So please don't forget that. Okay. So just give me 1 minute. But actually what happened is one fellow has come and he has taken this only. I spoke to customer care. Okay. They said that there is a problem from the Korean guy. I will speak to customer care. Sir, I will do this. Okay, sorry. Let's continue. Good morning everyone, good morning. Okay, so friends, let's quickly understand. In the last class we were started studying about the cancer. If every one of you very quickly recall, friends, we have clearly studied the characteristic of a cancer. And if every one of you remember, friends, in the last class we have completed till the process of what we have studied, till the characteristic, that is the till the metastasis we have completed, correct na? I hope every one of you remember. Friends, metastasis cannot occur very easily. In the process of metastasis, friends, first there is a process of invasion. After the process of invasion, what will happen, friends? The cancer cells has to move. That process, what do we call it as, friends? We will be calling it as the angiogenesis. After the invasion, after the angiogenesis only, friends, the process of metastasis is going to takes place. Okay, I will go little faster, friends. I am requesting all of you, please follow up because we have discussed it. So this is not the class where I am explaining depth of the concept. This is the class where we are doing the revision, so you have to follow in that way. Okay, so friends, what are the causes of the cancer? Follow me correctly. We all people know that very well, that friends, the cancer basically caused by the mutations in the cells. It was very clearly understand that the cancer is basically caused by the mutations in the cells. Now the answer is that, my friends, please listen my statements very clearly. These mutations can occur in the cancer because of any reason. It can occur because of a biological agents. It can occur because of a chemical agents. Friends, even it can occur because of a, what we can say, that radiations also. Radiations like UV radiation, high exposure to the X-rays, gamma rays, they can also cause the cancer. Certain chemical agents, especially friends nitrosamines, they can cause the cancer. When I use a word biological agents, all the students remember that friends, please keep it in your mind. Both bacteria also can cause the cancer and viruses also can cause the cancer. Everyone try to understand very quickly friends. When it comes to the radiation, all the students remember X-rays, gamma rays and other forms of radiations. Good morning Vinita. They can cause the cancer, but you have to remember that most of the people will say that if you use the cell phone for a longer time, because friends, this kind of general knowledge questions are very important in our exam. Using of cell phones for a longer period of time, it may also, good morning Saima, it may release the lot of radiation and it may also cause the cancer. But the point is very clear friends, till today there is a no documental evidence. What friends, till today we did not find any kind of a documental evidence to prove that the cell phone radiation causes the cancer, so we can't say that, okay. But friends, apart from this radiations, especially if you exposure to the more amount of the radioactive elements, especially friends uranium and especially friends thorium, okay, uranium and thorium, please remember, don't get confused, don't forget, uranium and thorium, they can increase the risk of the lung cancer. X-rays, CT scan, PET as well as the MRI, friends, please remember that these radiations never cause the cancer when you undergo the radiation 1 or 2 times. Because friends, the medical procedures are very small, but you have to remember one important point, that when you are undergoing these particular kind of scans in a periodic time, then only there is a chance that the cancer can occur. But when we do in the span of 6 months one time, in the span of 1 year one time, it never causes any kind of a cancer. So don't get confused and don't worry that I have to undergo the MRI or I have to undergo the chest X-ray or CT scan. We can, if it is necessary, of course we can take that particular scans, but periodic exposure to these kind of scans may increase the risk of cancer. Good morning Deepika, continue. So that is what radiations. And friends, when you understand the chemical carcinogens, here whatever the examples that I have kept in this particular slide, I am requesting all of you, please remember all the examples: asbestos, nickel, cadmium, radon, vinyl chloride, benzidine and benzene. You know, whatever the chemical substances that I kept here, all the students remember that they are all having what kind of activity they are having friends? They are all having carcinogenic activity. So friends, whether at the low concentration or at the higher concentration, all these substances are having the carcinogenic activity. That's why periodic exposure of all these chemicals surely will increase the chances of occurring cancer. Don't get confused. You can ask me a valid question. Sir, how all these chemicals are causing the cancer? You said that all these chemicals are having the carcinogenic property, so how they will cause the cancer? All the students look into this. They all can cause the cancer friends by altering the cellular metabolism. Once again understand, by altering the cellular metabolism or by damaging the DNA in the directly in the cells. So in one word I can say that all these chemicals will cause alterations in gene sequences, just because of the alterations in the gene sequences, which leads to the higher rate of mutations, which causes the cancer. And for our exam point of view, even it is for the part B also as well as for the part C also, what we people has to remember compulsory is friends, biological agents. Don't get confused with that point. All the students listen. Good morning Lokeshwari. 6 infectious agents. I don't know what you do, you have to remember it compulsory. 6 infectious agents have been shown to be carcinogenic primarily. by inducing a chronic inflammation. I want you to remember there are two viruses are having the carcinogenic property. What is that? Hepatitis B virus, hepatitis C virus. Please remember friends, and a bacteria, Helicobacter pylori. We all people know very well friends, Helicobacter pylori, what it will causes generally. Helicobacter pylori will causes the ulcers. If you don't treat the ulcers on the right time, the ulcers leads to what? Cancers. So that's why you can say that the Helicobacter pylori infection has to be treated on the time. If we delay the treatment, there is a great chance that it leads to the ulcers. At the same time, three worms such as Scytostoma hematobium, Abstioris viverni, and Chloronyx sinensis. I know these terms are little difficult to understand, but I am requesting all of you remember this 6 example: 2 viruses, 1 bacteria, and 3 worms. Now, what is the common point? Friends, follow me. What is the common point among all these 6 agents or all these 6 organisms? There are these 6 organisms having the carcinogenic property. That is the point I want you people to remember. Especially friends, all the students listen my words. Especially HBV, HCV and Helicobacter pylori. You know how they can show their mechanism of action. Especially HBV, HCV, they are responsible for causing liver cancer also. At the same time, they can also cause the breast cancer. They can also cause the prostate cancer also. They can cause the breast cancer as well as the prostate cancer also by acting on that particular hormones which are involved in the prostate gland development, and the hormones which are involved in the breast cells development. By inhibiting the growth of breast cells development, by inhibiting the growth of the prostate cells development, which leads to the cancer of a prostate gland as well as the breast. Now you have to remember this chart. I told you last time also, in 2020 November paper we got a same direct question in the part C just from this particular table. So I don't know what you do my friends, I want you people to remember it without fail. Virus, the tumors associated with it. You people no need to remember the area of a high incident or where it has occurred. It's not required at all. All the student, whatever I am marking, just remember as it is. Friends, when I stressed a particular point, when I requested you to remember, it is your utmost responsibility to remember that, because from there 90% of the chances are there that we can expect the question. At least we can expect one statement. That one statement also will help us to solve the question. So never neglect. Okay, now friends, DNA viruses, RNA viruses. See, sometimes you know what kind of question they can ask. They can give viruses in one column. They can ask in another column their genetic material. In addition, they will ask cancer caused by it. So 3 columns they will give only in one matching question, and they can raise the question. So that's why friends, you have to remember everything without fail. So what are the viruses that are having DNA as their genetic material? We all people know that 95% of the viruses they are having RNA as their genetic material. There is no doubt about it. But there are certain viruses. We don't need to read the viruses that are. We don't need to study the viruses which are having the genetic material as their DNA in the plants. We don't need to read. We have to majorly read about the viruses which are having carcinogenic property in our body. Okay, we have to read only with respect to the humans or animals. Papilloma virus. Papilloma virus will cause the wart also. And friends, please don't get confused. It also makes us confused. Papilloma virus, friends, there are different strains are there. So please don't get confused. Some of the papilloma virus, friends, they will normally cause the wart. Wart means what? It's a kind of a viral infection, which can form a bubble-like thing on the chest or on the stomach or anywhere on the body. But the point is here, friends, that is purely benign. It is not at all a dangerous one. Whereas I can use the word, friends, 80% of the infections caused by the papilloma virus, mostly they are the benign only. They are not too much dangerous. Only 20% of the papilloma viruses, they can cause the infection, friends, which are purely cancerous. They can cause the cancer of a uterine as well as the cervix also. I want you to remember it without fail. Then hepatitis B virus, we already studied that, which can cause the liver cancer. Sometimes instead of using the word liver cancer, they will use the word hepatocellular carcinoma. Hepatocytes means the cells of the liver. So if the cancer occurs specifically on the hepatocytes, we can call it as the hepatocytocellular carcinoma, which is exclusively caused by the hepatitis B virus. Then friends, Epstein-Barr virus, don't get confused. One of the most dangerous cancer, and the people who get affected with this particular cancer, their lifespan will become very low. Any person who get affected with the T cell cancer as well as the B cell cancer, friends, their lifespan is extremely lower even compared to the HIV. The people who are having the HIV, they can live very healthily by taking proper medicine up to 5 to 15 years, and most of the people are living more than 25 years after their infection also. But the cancers which are associated with the B lymphocyte and T lymphocytes, friends, the chances of survival is very, very low. Epstein-Barr virus, which is causing the Burkitt's lymphoma. Burkitt's lymphoma is a type of cancer of B lymphocytes. Don't get confused with that particular point. Now, retrovirus family, human T cell leukemia virus. Right. So, it basically causes cancer in the T cell, especially on the T helper cell and the T cytotoxic cells. We all people know very well. Cytotoxic T cells are those type of cells, they will only kill the cancerous cells. But now you try to understand, the cytotoxic T cells only got damaged now. They only got tumorized. Hence the immune system become extremely weaker and weaker and weaker. Now next one, HIV virus. Friends, please never ever confused, okay? HIV virus never ever cause the cancer. That is absolutely wrong statement, okay? HIV virus, what it will do? We all people know that it can suppress the immune system. Correct, na? You also know very well it can suppress the immune system, especially by blocking the T helper cell and very specifically CD4 receptor. We all people know about it. Now the question is, whenever our T helper cells and CD4 receptors get completely suppressed or inhibited, what is going to happen? Every one of you listen to me. What is going to happen, friends? Immediately our entire immune system get compromised. Our immune system get compromised. Friends, whenever our immune system get compromised, what is going to happen? Because of the compromising of immune system, our immune system is unable to produce the antibodies. It cannot fight with any kind of infections, including the HIV person. When they enter into the AIDS condition, even they can't tolerate the common cold. They can't tolerate the common cough also. Now this time, there are certain pathogenic organisms. We will be calling them as the opportunistic pathogens. What we will be calling them as? We will be calling them as the opportunistic pathogens. That opportunistic pathogen speciality is what? If the immune system is stronger, they can't do anything. When the immune system becomes weaker, that opportunistic pathogens start acting. Now, what is the example of that opportunistic pathogen? Kaposi's sarcoma virus. And what is the speciality of this Kaposi's sarcoma virus? it is having a carcinogenic activity. So please don't confuse. HIV virus never caused the cancer, but the HIV virus suppress the immune system. By that it is helping, it is allowing other pathogens to enter in our body, which is having carcinogenic activity. Don't get confused with that. I don't know what you do. It's your responsibility to remember everything without fail. Now friends, so far till this slide we have understand characteristics, properties, and most important causes of the cancer. If you don't get the question from the characteristics or the causes, there is a great way of asking the question, that is from the mutations. Most of the time a difficult, I don't use the word difficult, I can use the word tricky question. Okay, and keep it in your mind that tricky question also purely statement based question. Okay, so now try to understand what kind of mutations that are causing the cancer. See friends, please don't get confused, follow me correctly. Many genes are altered in the human cancer. Friends, I want every student has to remember one unique statement. What is that? In the yesterday class I told to all of you, friends, there are more than 40,000 types of cancers are there. What I told to all of you, there are more than 40,000 types of cancers are there. Now what's the interesting point? Among 40,000 type of cancers, what will be the one common characteristic? In every cancer, the common characteristic is, or the common cause, the common cause is cancer is occurring because of the alteration in the genes, alteration in the nucleotide sequences. The reason may be anything. The reason may be biological agent, the reason may be chemical agent, the reason may be radiation, doesn't matter. But what is the common strategy, sorry, the common point here? In every type of a cancer, the mutation is occurring just because of the alteration in the gene. So every student keep it in your mind, one of the major reason for the cancer to occur is change in their gene sequences. This is the major point. Many genes are altered in the human cancers. So friends, so far, or you can say till today, we have identified 100 types of critical cancer genes. 100 type of critical cancer genes were identified so far. And my friends, the question is very clear. From here every student pay attention. It is important for the part B, more especially for the part C. All these 100 type of critical genes we have categorized simply into two types. One we can call proto-oncogene, another one we can call tumor suppressor genes. Now you can ask me a valid question, sir, based on what we categorized 100 cancer genes into just two types, based on what criteria? Whether they are causing the cancer, you have to follow me very correctly, whether they are causing the cancer because of a too much activity, or whether they are causing a cancer because of a too less activity, because of a too much of activity or because of a too less activity. In other words, I can use the word, it is causing the cancer because of a gain of function of a gene or a loss of function of a gene. Always remember, these are all the two major criteria that we will consider all the time while categorizing the tumors. So friends, pay attention. And what you have to remember, the movement we already read, so that's why I'm explaining exactly what you have to remember. I'm not going too depth. I'm explaining exactly what you have to remember during the time of a revision. Follow me correctly, remember that only. The moment you heard the name of a proto-oncogene, what's the first point you have to remember? Proto-oncogene always show dominant mutation. The first point you have to remember. Okay, and the moment you heard the name of a tumor suppressor gene, what you have to remember? Tumor suppressor genes also caused by the recessive mutations. Okay, my friends, remembering these 2 points are utmost important. Do not get confused with this particular point. I will explain both very clearly. You people has to know the concept, that's all. However, they may ask the question, how tricky they may ask the question that, however, they may ask the question, how tricky they may ask the question, concept is same. If you understand the given question correctly and if you know the concept correctly, you can answer it very easily. So pay attention. First we are talking how a proto-oncogene converted into oncogene. First they will give one statement. See, I am giving one statement to all of you. All genes in our body called as proto-oncogenes. I have given you a statement that, friends, all genes in our body called as the proto-oncogenes. Now the question is, do you think in our body is there any gene named as proto-oncogene? Do you think is there any specific gene called proto-oncogene? No. In our body there is no gene as such as proto-oncogene. Listen my statements carefully. Any gene, friends, focus, any gene which is working normally in our body, we will be calling it as the proto-oncogene. Any gene which is working in our body normally, we can call proto-oncogene. Now, any gene which is working normally, we are calling proto-oncogene. Now, why this proto-oncogene is converting into oncogene? That's the valid question. Now, the answer is, my friends, we all people know that proto-oncogenes having one particular function. An example, there is a gene. This gene having a specific function. Whenever the gene is performing its specific function, there will be no problem at all. But you know what happened? This gene gain an extra function. This gene gain an extra function. Actually, whatever the function it got gained, it is not actual function performed by that gene. So what is happening? Because of that particular extra function, the gene can undergo the mutation at a very higher at a faster rate, because it is gaining an extra function which it can't perform actually. That's why it undergo the process of mutation. Now I have used one unique statement. What is that statement I have used? Friends, proto-oncogenes always can show the dominant mutation. What do you mean by that, friends? You can see the picture. This is the normal cell. Correct, na? All the students listen. How many copies of genes are there? This is the first copy of the gene. This is the second copy of the gene. Now the point is very clear, my friends. Since it is a dominant mutation, friends, the question is very clear. How many copies of the genes got mutated? All of you please look into the diagram. Only one copy of the gene got mutated. This is a unique statement everyone has to remember. Only one copy of the gene got mutated. But what's the point? Even though one copy of the gene got mutated, that is sufficient to convert this proto-oncogene into oncogene. Why? The reason is it is a dominant type of mutation. Never ever forget this particular point. And friends, when you look into the underactivity mutation, what do we call it? This as loss of function mutation. Don't get confused with this particular point. See, this is what? Tumor suppressor gene. Every student has to remember the point that tumor suppressor gene always show loss of function mutation. And very important is what, friends? The type of mutation that it show is the recessive mutation. Please don't get confused. Now all the students pay attention without confusing here. So now first of all, you can ask me, sir, what are the functions of a tumor suppressor gene? Do you think is there any gene called as a tumor suppressor gene is present in the body? Of course, friends, there is a gene in our cell. There is a gene called as tumor suppressor gene. Name itself indicating it is going to suppress. What it is going to suppress? It is going to suppress the formation of a tumor. That's why we are calling it as the tumor suppressor gene. And the point is very clear. Don't get confused. You should not think that tumor suppressor gene is active every time in our body. That is wrong statement. They can give a statement that tumor suppressor genes are active all the time, which is a wrong statement. Tumor suppressor genes become active whenever there is a damage in the DNA. Whenever there is a change in the sequences, then it will become active. But all the students look into the speciality of a tumor suppressor gene. Since it will show the recessive mutation, how many copies of genes are there, friends? There are 2 copies of genes are there. Friends, even though 2 copies of genes are there, out of 2 copies of genes, everyone pay attention, 1 copy of the gene got mutated. What, friends? 1 copy of the gene got mutated. Do you think the tumor suppressor gene lose its function? Never ever. Why? Because it is showing a recessive mutation. Since it is showing the recessive mutation, one mutation never ever because of one mutation it never lose its function. So see here, there is a single mutation occur, no effect of mutation in one gene copy. That is sufficient to work normally. Now you only look into this, friends. There will be a second mutation also occur. Because of the second mutation, what is happening, friends? It is going to inactivate the second gene copy also. Don't get confused with that particular point. Now friends, we call it as a recessive mutation, because if the mutation wants to occur, friends, there should be compulsory 2 mutations has to occur in the tumor suppressor gene. And why the tumor suppressor gene is causing the cancer? Because, my friends, tumor suppressor gene is losing its function because of loss of function. Never forget. At the same time, here the mutation is recessive. This is only you remember. In case of a proto-oncogene, it is the dominant type of mutation, and the most important point you have to remember is that here single mutation is sufficient to convert into cancerous. Please focus, never get confused over there. Now what we do is we will study. What we study? We will study how proto-oncogene converted into oncogene. Just now I have explained how it will happen. Let's take certain examples. Later we will study how tumor suppressor genes are losing its function and how they are causing the cancer. Apart from this, there is no way of asking the question. So focus. Every one of you friends pay attention. I am going little speed, but I hope you are following me correctly. Proto-oncogene, how it is getting converted into oncogene. One point is very clear, right? We clearly studied that. Friends, any gene can cause the cancer because of alterations in that particular gene sequence only. Now you only try to understand what kind of alterations that are occurred in the proto-oncogene. Keep on understanding me. There are n number of changes can occur. See here, there is a deletion or point mutation occur. But I have written one word here. Friends, suppose example, this is a non-coding region and this is a coding region. Suppose example, friends, in the non-coding region, an example, 2 bases or 2 nucleotides got removed. It won't cause a greater mutation at all. I can use the word that it will cause the silent mutation. It is having a negligible, follow me correctly, it is having a negligible effect on the function of a gene. But even though, every student remember, even though there is a single mutation occur in the coding region, it will change the entire function of a protein. Okay, which is a very dangerous mutation. So a proto-oncogene, because of a deletion or a point mutation in a particular coding sequence, it will undergo the mutation. I am not at all using a word that it cannot synthesize the protein. I am not saying that. It can synthesize the protein, but the point is here, the protein, there are many possibilities. The protein become hyperactive, or the protein gain an extra function. Hyperactive or it gains an extra function. But what is the point? Protein is getting synthesized. So hyperactive protein made in normal amount. So this is one kind of a reason why the proto-oncogene converted into oncogene. So next one, gene amplification. You all people know that our gene should not replicated continuously. You know very well how many number of times it has to be replicated. Only that many number of times it undergo the replication, then it will undergo the transcription. Finally they will synthesize the protein which is required. But what is happening to the proto-oncogene? Because of a mutation, what is happening? The gene is mutation only, multiple amplifications. It is amplifying, it is multiplying unnecessarily. As a result, what is happening, friends? The normal protein is greatly overproduced. Do not get confused with that particular point under any circumstances. Next one, chromosome rearrangement. This is so. What is the statement they will give in the 4-marker question? Proto-oncogene converted into oncogene because of the chromosome rearrangement. Most of the students will get super confused with the statement. First of all, what do you mean by the word rearrangement of the chromosome? Are we deleting any sequences? No. Are we adding any sequences? No. We are just rearranging the sequences without deleting, without addition. Now listen. We have studied many times that, friends, every gene has to be regulated by certain regulators in the cell. So the red color one is the gene. It is going to be regulated by this particular green color gene, okay, which is extremely important for the process of regulation. Suppose example, this regulatory gene got failed. This regulatory gene got failed. If the regulatory gene is working, this protein never be overproduced. How much our concentration of this protein has to be produced, it will synthesize in only that amount. But the regulatory sequence or the regulatory region got mutated or it is not functional, then what is happening? This protein will be synthesized in the very higher amounts. Do not get confused with that particular point. Now, what's the unique statement you have to remember? Friends, focus. Once again, try to understand. This is the DNA and this is what, friends? This is the gene segment of another gene. Suppose the yellow color one is the different gene and the green color one is the different gene. What is happening? There is a fusion occur. There is a fusion or there is a joining occur between the two different genes. As a result, two things will happen. What will happen? This gene may influence the function of this gene or this gene may influence the function of that particular gene. So as a result, what is happening? Suppose gene 1 influence the function of a gene 2. Whatever the protein that is coded by the gene 2, it becomes hyperactive. Similarly, if the gene 2 influence the function of a gene 1, whatever the protein that is synthesized by a gene 1, it will also become hyperactive. Whatever the reason it may be, friends, whether it is a deletion or a point mutation, whether it is a gene amplification, whether it is the chromosome rearrangement, because of all these reasons, my friends, the proto-oncogene is getting converted into oncogene. Please don't get confused. Now, let me give you one wonderful example of this particular point mutation. I want you to remember it. Okay, friends, all the students follow me. Proto-oncogenic receptor. Now you have to remember, when I use a word proto-oncogenic receptor, what do you have to remember? My friends, in our body, any surface receptor, if it is working normally, we can call it them as the proto-oncogenic receptor. There is no doubt about it. Okay, any receptor, if it is working normally, functioning properly, we can use the word proto-oncogenic receptor. Now listen, friends, this particular proto-oncogenic receptor, it is converted into oncogenic receptor. What will be the reason? I told to all of you, the reason is very simple, just because of a point mutation, just there will be a change in one nucleotide, that's all. Now friends, this is what you all people know, right? We studied this receptor having three domain: extracellular domain, there is a transmembrane domain, and you can see the another domain, we can call it as the cytosolic domain. Correct now friends? There will be three type of domains are present. Now every student remember this transmembrane domain, in the active site there is a presence of amino acid valine. It is present in the transmembrane domain. Because of a point mutation, what has happened, you only look into this. Because of a point mutation friends, valine converted into glutamine. The moment valine converted into glutamine, now every student has to remember. We have studied in the cell signaling topic multiple times. What we studied? Friends, without a ligand, receptor never bind with each other. Without a ligand, receptors will become monomers. Upon ligand binding, the receptor become dimerized. Then only they will continue the process of cell signaling. That is what we people studied. Now what you have to remember? You have to remember a point that because of a point mutation, focus friends, even though there is a no ligand, even though there is a no ligand, even though there is a no signal, even though there is a no primary messenger, it can easily dimerized. It will dimerized, undergo the process of autophosphorylation and continuously passing the signal, obviously, which may leads to the tumor. It's a very simple, because friends, without ligand only the response is happening. Why the response is happening? Suppose example, a particular ligand is responsible for cell division. Okay, now the ligand only not there, the cell is dividing uncontrollably even in the absence of a ligand, obviously leads to the cancer. So what I would like to conclude here, proto-oncogenic receptor can be converted into oncogenic receptor and causes the cancer just because of a point mutation or a deletion of just a single base or a deletion of one nucleotide. So these are all the ways how the proto-oncogenes can be converted into oncogenes. And friends, from where we can expect most of the time the questions are tumor suppressor gene only. Either in the part B also friends, or in the part C we can expect the tumor suppressor gene questions. Now first of all, they can ask a part B question that, listen my questions, listen my statements carefully, because you will get only statements. All of the following functions are performed by the tumor suppressor genes except, listen to me carefully, the statement are like this: all of the functions are performed by the tumor suppressor gene except. In other words, to make us confused, they can ask a statement that which of the following function cannot be performed by the tumor suppressor gene. Like this they can ask the statement. Otherwise, which of the following function cannot occur in the absence of a tumor suppressor gene. So in a different way they can ask the statements. So first what you have to remember, what are the functions of the tumor suppressor gene in a cell, that you have to remember. Keep it in your mind, tumor suppressor gene, which is going to suppress the tumor formation. I can tell you friends, they will do majorly three functions. First of all, I also told to you friends, tumor suppressor genes are not going to be activated every time. They are going to be activated whenever there is a DNA damage. Correct na? Whenever there is a mutation, then only they will be activated. What they are going to do? The three major functions performed by the tumor suppressor gene. It will stops cell cycle, first function. Because once it identifies the damage, we will see how it will identify the damage. First it stops the cell cycle, then it immediately, everyone remember, it immediately stimulates, it immediately activates DNA repair mechanisms. It will activate the DNA repair mechanisms. 90% of the cases, tumor suppressor genes will activate the DNA repair mechanisms. By that damage can be repaired easily. In worst case, if that particular damage is not repaired, what will happen, friends? Obviously, there is a process of apoptosis occur. What will occur? There is a process of apoptosis occur, that is programmed cell death. What's the point you have to remember here? What is the conclusion I want to say? If, listen my statements carefully, if tumor suppressor gene is present, even though damage occur, even though mutation occur, tumor suppressor gene make sure that it will try to repair it. If not, it will kill that particular cell, but it make sure that that information never passes to the next generation. That is the speciality of a tumor suppressor gene. Tumor suppressor gene never allow the growth of a tumor till it is active. That's an important job of this particular tumor suppressor genes. Now, what are the various type of the tumor suppressor gene? There are many tumor suppressor genes are there, friends. The first and major type of tumor suppressor gene is PRB. The first identified, don't get confused, the first identified or the first discovered tumor suppressor gene is the PRB gene, protein retinoblastoma gene. Then most widely studied in the history of a cancer, and still a wonderful research is going on in the cancer field exclusively based on a tumor suppressor gene P53. This is the most important gene among all. Then followed by the P21, you have to remember all. Then followed by the P16. Then friends, even you can take an example of TGF, transforming growth factor. Even you can take the sixth example as BRCA1 as well as BRCA2, breast cancer genes, right? So I have given the 6 examples. Please remember all the 6 examples without fail. Among the 6 examples, what you people has to study mostly for the exam? You have to study the PRB, you have to study the P53, you have to study the TGF, and you have to study the BRCA1 and BRCA2 and how they will work. Okay, one point is very clear. What's the ultimate function of the P53? I also told you that P53 is the most important. P represents, friends, protein, that's all. Tumor protein 53. Okay, 53 will be, you can say that molecular weight. You always remember it is the P53. Now please remember, what is the statements that they have asked in the exam which most of the students won't understand properly. Friends, you follow me correctly, listen my statements correctly. P53 is the tumor suppressor gene. I have told to you that it blocks the cell cycle or cell division, activate the DNA repair mechanism, and stimulate the process of apoptosis. Correct na? Now what is the question they asked? They have asked a question in the form of a statement. P53 is a nuclear transcription factor. The first statement. And the second statement, they asked that P53 can cause P53 can show pro-apoptotic function. And the third statement they asked 50%, this is the valid, wonderful statement. What is the third statement? 50% of the human cancers, 50, I'm using the word 50%. 50% of the human cancers are occurring because of mutations in the P53. These are all the 3 statements they have given in the exam. All the 3 and what is the fourth statement? The fourth statement they asked that P53 is not active all the time. It is always not active. Friends, 4 statements are absolutely true statement, because you have to remember a point. Why the tumor suppressor gene has to be activated every time if there is a no damage, if there is a no mutation, if there is a no error? Why should we activate it? So it causes a lot of consumption of the energy. So we don't activate it every time. Whenever there is a damage occur, then only P53 levels will be raised. P53 level will be raised. So The statement P53 is not active always, right statement. Second point, P53 is a nuclear transcription factor. Every student will say that how it will become a transcription factor. The answer for this particular statement, focus, follow me correctly. P53 by acting as a transcription factor, follow me friends. P53 by acting as a transcription factor, it is stimulating other tumor suppressor genes like a P21 and P16. If the P21 and P16 wants to be activated, they require a transcription factor. So what will be the transcription factor for these genes? P53 itself. Don't get confused. And P53 having a pro-apoptotic function, and 50% of our human cancers are exclusively occurring because of the mutation in this particular P53 gene only. Don't get confused with this particular statement. You never get a difficult question. You will get a statements which are super confused. Okay, they are not confused, they are tricky. You have to know that correctly. Now listen friends, all the students understand there is a damage occur. Now you have to listen. I have written a point that when the damage occur, which level becoming rising? P53 level becoming rising. Now the question every student has to remember, how does the P53 level will rises? This is the question. Keep it in your mind. If the P53 level wants to be raised, there will be a compulsory, the process of phosphorylation has to occur. This is a unique point friends. These kind of statements are very important. P53 concentration wants to rise in our body, it should undergo the process of phosphorylation. But the point is very clear, it never undergo the process of phosphorylation all the time. It will undergo the process of phosphorylation only when there is a damage. Upon damage, P53 level will be rises, and friends, P53 immediately arrest the cell. Of course, it has to arrest first. After arresting, what it will do? It never kill the cell immediately, okay? It will always try to repair it before it undergo the division with the help of repair mechanism. If that is not possible, then only it will kill the cell. Always remember that. But you only try to understand same condition. DNA got damaged, but what happened? P53 gene got mutated. Friends, if a P53 gene got mutated, do you think the damaged cell get arrested? No arrest. If there is a no arrest, it will undergo the division. It will undergo the division uncontrollably. So remember, uncontrollable division with damage. See, understand the dangerous condition here. Uncontrollable division with a damage. As a result, my friends, obviously it can cause the tumor formation. Right? So friends, there is a very little part is left. We will discuss in the afternoon class. Okay, hardly in 10 minutes I will finish this concept. We will move on to the cell signaling pathways. Okay, I don't teach all the signaling pathways. I will cover major important pathways very fast. You also don't read everything. We don't have much time. Okay, read with respect to the revision. Okay, I hope everyone understand the concept. If you have any doubts, please ask me. And I'm requesting you friends, please study minimum 10, maximum 13 to 14 hours. Okay, and sleep peacefully for 6 hours. Sufficient. Okay, sleep peacefully for 6 hours. If you have really having a patience, try to read more than this, because friends, I strongly believe last 7 days are very important to recall whatever we studied, because if you remember whatever we studied in the examination hall also, you can recall it very faster. Okay friends, that's all for now. I'll meet you at a sharp 3 p.m. Okay, bye bye.